That's why HIV infects helper T cells, because helper T cells are the ones that have CD4 on it. Okay. Right, and so that acts like a docking point from which the virus can be drawn toward the host cell, and then from there it can fuse with the host cell and introduce the nucleic acid into the T cell. Okay. All right. So think about this: if you could prevent GP120, right, from interacting with CD4, what would happen to the virus?
Die.
Yeah. Would it be able to infect? No, right? So it wouldn't be able to attach, right? Okay. So one therapeutic target is to try to mess up this interaction. Okay, and so researchers have made different attempts to do this, either by finding a drug that can do that, or by developing a vaccine. If you can develop a vaccine that could a vaccine that would allow your immune system to produce antibodies that have a strong attachment, strong binding to GP120. Okay, think about that. Block up all the GP120, right? That's going to prevent it from interacting with CD4. Does anyone remember what we call that? If you have a virus and you bind it all up with an antibody, what's that called? So here's your virus.
Is it neutralization?
Yeah, neutralization. Good. Okay, so that's neutralization. So that's what strategy involves. In fact, that's what's still being done. It's trying to figure out a vaccine that can actually neutralize the virus, just like this. Okay. All right. So turns out that this simple model doesn't work, but there are some other proteins on the virus that might be potential targets. And so that's what's being pursued now. Any questions about that? So this gets into reverse transcription. Right. So the question is, how does the HIV genome replicate? Right. So we start with RNA. So how does replication occur since we're doing reverse transcription? So remember that HIV has two identical copies of single-stranded RNA. That's its genome. It uses an enzyme called reverse transcriptase. And what that enzyme does is that it makes double-stranded DNA from that RNA, from that single-stranded RNA. And that's shown simply here, where we have, here's our single-stranded RNA. That gets reverse transcribed by reverse transcriptase. That enzyme has the ability to make a copy of that RNA, but now that RNA copy is a DNA copy. Okay? So the RNA, it uses this RNA as a template to make a DNA copy. Okay? Then what it does, and this looks more familiar to us, it just takes that single-stranded DNA molecule and makes a copy. Use that as a template to make the second strand of DNA. Okay. Now you have double-stranded DNA, and that double-stranded DNA can insert into the human chromosome. Right? So remember, the human chromosome is DNA, and so this viral genome is now DNA. So it can take that DNA and simply hide inside the host chromosome DNA. Right? And that's what happens. Okay. Any questions about that? Right, so the enzyme that does this, that integrates that double-stranded DNA into the host chromosome, that's called integrase. Okay, integrase. All right, so we have two enzymes. One is reverse transcriptase to do this process. The second is integrase to do this process. Okay. Any questions about this? Once it's inside the host, then this actually is used to make more viral RNA, and that viral RNA becomes part of the new viruses that are produced from this infection. All right, now there's another protein that's important that that was shown on the picture of HIV. So I wanted to spend a little time talking about that, since this also has some clinical significance. Oh yeah. So speaking of clinical significance, if you could design an inhibitor of integrase, right, or an inhibitor of reverse transcriptase, what would happen to this process? Yeah, would you be able to make more virus? No, right? Because you basically put a stop to it by interfering with one of these enzymes. So in fact, there are reverse transcriptase inhibitors. There's also integrase inhibitors. Okay, so there are drugs that act in that fashion. They try to inhibit reverse transcriptase, or try to inhibit integrase. All right. So another enzyme that's involved in producing more HIV. So again, this is another therapeutic target. Right, so we developed another therapy based on this enzyme, on this protein. It's called HIV protease. HIV protease is involved in processing the proteins that HIV needs to make more virus. So what happens during HIV synthesis, or biosynthesis, okay, is that the protein has to be made. The protein that's going to be part of the HIV virus. The protein gets made as one long chain, okay? Right, so it's just like if you want to build a piece of furniture, you buy this one big piece of wood, and then you have to cut it up into the small pieces to make your piece of furniture. Okay? That's what HIV does with this protein. It has a protease. Okay? That protease breaks down the protein into smaller pieces, right? So it actually doesn't destroy the whole protein, it just cuts it at certain spots, like we have here. And those smaller pieces are used to make the new virus. Okay? All right, so you can see how that process occurs here. Now, there are drugs called protease inhibitors. Protease inhibitors. So here's a protease inhibitor, and what that does is that blocks the protease. Okay? All right, so that inhibits the protease. And so what's going to happen to this protein? If you block the protease, what's going to happen to the protein? Is it going to get cut? No, right? If it doesn't get cut, can you make more virus? No, right? Okay? See that? All right, so that's another therapeutic strategy. And in fact, there are protease inhibitors. Okay? So if you've heard of protease inhibitors, that's what they do. And they inhibit this step, they inhibit this enzyme to prevent more virus from being made. Okay? All right, so we have these variety of different potential targets that we can use to treat HIV. Okay? And there are some that already exist. So based on that, there's a type of therapeutic strategy that's used now for HIV. And this came out several years ago. It's still being used. It's called HAART, H-A-A-R-T, for highly active antiretroviral therapy. Okay. All right, what this is a term for is simply it's using multi-drug therapy. Right, so you use a combination of drugs to try to control HIV. Okay. All right, so in one formulation, you can use 3 different anti-HIV drugs. One would be a reverse transcriptase inhibitor. Okay. So remember, we talked about one type of reverse transcriptase inhibitor. It's called AZT. Okay, so AZT is one of the first big anti-HIV drugs. That's a reverse transcriptase inhibitor. Okay. How this inhibitor works is that it gets incorporated into the viral DNA during reverse transcription. So it puts in this nucleotide analog into the viral DNA, and then that prevents the DNA from being used. Okay. There's also non-nucleoside reverse transcriptase inhibitors. These are direct inhibitors. These just directly bind reverse transcriptase. Okay. And then the third one are protease inhibitors. Right? That prevents viral protein processing. Okay, so HART involves combining 3 drugs from these 3 different categories. Okay. Now there's more categories that we can use, but here are the initial 3. Right? We have to have 3 different categories so that, actually, does anybody know why? Why can't we just use one drug? What would happen so if we just use AZT, what would happen over time? Okay, it could change, right? But what else? So think about this just in terms of drugs in general. Like, let's say this was a bacterial infection, and we used one antibiotic.
Get resistance?
Yeah, you get resistance, exactly, right? And that results from mutation, right, of the virus. But it's able to resist the drugs. The drug no longer works against it. And this is what happens with HIV as well. Okay. So we use more than one drug at the same time, it's more difficult for the virus to develop resistance because now it has to battle 2 drugs or 3 drugs. Okay. Any questions about that? So something to keep in mind is that there have been studies done on HIV patients to try to figure out how many different mutations or how many different types of HIV virus can you find in a patient. And it turns out to be some incredible number. When they actually looked at the genetic sequence for each, for all the different types of HIV they could find in a patient, it was something like 500,000. All right, 500,000 different combinations of nucleotides to make up an HIV virus. All right, so that gives you it's like buying 500,000 Powerball tickets. Okay, that really increases your chances of finding that winning combination, at least from a viral point of view, to resist those drugs. Right? That's what makes it so difficult to develop a way to get rid of this virus. Any questions about this? Let's see if I can. I think I can show you this now. I'm gonna have you do a worksheet a little bit early. So I'm gonna pass this